Hello, and welcome back to the Sleep Lab. Tonight's title is a little dramatic, and it's worth being honest about that before we drift any further. No single plant is waiting quietly in a field somewhere, poised to end civilization overnight. What we're actually going to explore tonight is something considerably more grounded, and in its own way more genuinely fascinating: the surprising, well-documented fragility hiding underneath humanity's relationship with the plant kingdom, the handful of historical moments when a disease, a blight, or a quietly spreading fungus came remarkably close to reshaping entire nations, and the ongoing, present-day vulnerabilities agricultural scientists still work quietly to defend against every single year. Civilization has always rested, more precariously than most of us ever pause to consider, on an extraordinarily small handful of plant species, grown at an extraordinary scale, in ways that leave the entire system more exposed to catastrophe than its everyday abundance would ever suggest. Tonight we're going to walk slowly through 13 genuine, historically grounded facts about the plants, blights, and agricultural vulnerabilities that have shaped, and could still shape, the fate of entire civilizations, told the way history deserves to be told, unhurried, calm, and considerably less frightening once properly understood than the title tonight might have first suggested. It's worth being honest about why a title like tonight's works so effectively in the first place, since understanding that honestly helps frame everything that follows more usefully. Civilization's relationship with plants sits in a genuinely strange psychological blind spot for most people, invisible almost entirely by virtue of its overwhelming success. Grocery store shelves remain reliably full. Coffee, chocolate, bread, and wine remain reliably available, year after year, decade after decade, an abundance so consistent and so unremarkable in daily experience that it becomes almost impossible to intuitively grasp just how much careful, continuous agricultural science labors quietly behind that abundance, holding back threats most of us will simply never have reason to hear about, precisely because the scientists and breeders working on them succeeded. Tonight's exploration isn't really designed to introduce genuine new fear into that quiet reliability. It's designed to offer a small, appreciative glimpse behind it, at the fascinating, largely unseen history and ongoing effort that has made such extraordinary, everyday abundance possible in the first place. If this kind of journey is worth your time, a like, a subscribe, or a thought left below helps this small lab keep its lights on for other sleepy minds finding their way here. But for now, there is nothing you need to do except settle in, let your body grow heavy, and let the quiet, surprising fragility of the green world drift gently over you instead. Let's begin. In the autumn of 1845, farmers across Ireland began noticing something quietly, terribly wrong with their potato fields, leaves blackening and curling within days, and the tubers themselves, once dug up, revealing a rotten, foul-smelling mush entirely unfit to eat or store through the coming winter. The culprit, later identified as a water mold called Phytophthora infestans, spread with devastating speed through Ireland's potato crop, a single agricultural species upon which an extraordinarily large proportion of the country's rural population depended almost entirely for basic caloric survival. What made this particular blight so catastrophic wasn't simply the disease itself, but the almost total lack of genetic diversity within Ireland's potato crop at the time, with the vast majority of Irish farmers growing essentially a single variety, called the Irish lumper, meaning the entire agricultural system carried virtually no natural genetic resistance anywhere within it, a single vulnerable variety planted across millions of acres, offering the spreading blight an almost uninterrupted path of susceptible plants stretching across the entire country. Over the following years, what became known as the Great Famine killed approximately 1 million people through starvation and related disease, while driving roughly another million to emigrate, primarily to North America, in a mass exodus that permanently, dramatically reshaped Ireland's population and left lasting demographic consequences still measurable today, with Ireland's total population only recently, in the 21st century, beginning to approach the levels recorded before the blight first struck. Agricultural historians studying the famine's underlying causes emphasize that the tragedy resulted not simply from the fungus itself, a naturally occurring pathogen present in South America for millennia without producing anything remotely resembling this scale of catastrophe, but specifically from the dangerous combination of genetic monoculture and a rural population left with virtually no alternative food source to fall back on when that single, vulnerable crop failed catastrophically and completely across an entire growing season. The famine's political and economic dimensions add genuine, important nuance to this agricultural story, since historians studying the crisis emphasize that the blight itself, while genuinely devastating, does not fully explain the staggering scale of resulting human suffering on its own. Ireland, at the time under British rule, continued exporting substantial quantities of other food crops, including grain and livestock, throughout the famine years, food that could have meaningfully alleviated the crisis had it been redirected toward the starving rural population rather than continuing to be shipped abroad under the prevailing economic policies of the period. A historical detail that has made the Great Famine a subject of enduring, serious historical and political debate, extending well beyond simple agricultural pathology alone. This dimension of the famine offers a genuinely important broader lesson relevant to nearly every historical crisis explored throughout tonight's episode, that agricultural vulnerability, however severe, rarely translates into genuine human catastrophe through biology alone, but typically requires some additional failure of political, economic, or logistical response layered on top of the underlying agricultural crisis to produce the kind of devastating, large-scale human consequence the Irish famine ultimately became. For much of the 20th century, the dominant banana variety sold and consumed throughout the world was called the Gros Michel, prized for its exceptional flavor, its resistance to bruising during long-distance shipping, and its reliable, easy cultivation across vast, uniform plantations throughout Central America and beyond. Beginning in the early 1900s, a soil-borne fungus researchers named Fusarium wilt, more commonly called Panama disease, began spreading through these plantations, and because the entire global banana export industry relied almost exclusively on this single variety, propagated not through seeds but through clone cuttings that left virtually no genetic variation anywhere across the plantations, the disease spread with devastating, unstoppable efficiency, ultimately rendering the Gros Michel commercially unviable across most of the world's major banana-growing regions by the middle of the 20th century. The banana industry survived this catastrophe only by switching almost entirely to a different variety, the Cavendish, which happened to carry natural resistance to that particular strain of Panama disease, and which remains, even today, the dominant banana variety found in grocery stores worldwide. Here is where tonight's fact becomes genuinely unsettling rather than simply historical, since the modern Cavendish banana is, once again, grown almost entirely as a single, cloned variety across the vast majority of the world's commercial banana plantations, meaning the exact same underlying vulnerability that doomed the Gros Michel decades earlier remains essentially unchanged today, simply relocated onto a different variety. Beginning in the 1990s, and spreading considerably more widely in recent decades, a new, considerably more aggressive strain of Panama disease, called tropical race 4, has begun infecting Cavendish plantations across Asia, Africa, Australia, and, as of relatively recent confirmed cases, Latin America as well, and because this new strain has proven resistant to the very same natural defenses that originally saved the Cavendish from the Gros Michel's fate, agricultural scientists studying the global banana industry consider it a genuine, active, ongoing race against time to develop a resistant replacement variety before this modern strain eventually, potentially, spreads widely enough to threaten the global banana supply in a way genuinely comparable to what happened to the Gros Michel generations earlier. Researchers working on this modern breeding challenge face a genuinely difficult technical obstacle worth understanding, since bananas, much like the cacao trees discussed later tonight, are typically propagated through cloning rather than conventional seed-based breeding, largely because commercially cultivated bananas are seedless, a trait consumers have long favored for easier eating but one that considerably complicates traditional plant breeding techniques relying on the genetic recombination that ordinarily occurs through seed production. This has forced researchers pursuing disease-resistant banana varieties toward more technically demanding approaches, including genetic modification and careful, painstaking crossbreeding using rare, seed-producing wild banana relatives, efforts that have shown genuine promise in recent years but remain, as of tonight's recording, still working toward a commercially viable, widely deployable solution capable of fully replacing the vulnerable Cavendish at the scale the global banana industry currently requires. Wheat provides, by most agricultural estimates, somewhere around 20% of all calories consumed by humanity worldwide, making it one of a small handful of crops upon which the vast majority of the global population depends, directly or indirectly, for basic nutritional survival, a dependence that makes any significant threat to global wheat production a matter of genuine, serious concern among agricultural scientists and food security researchers worldwide. In 1999, researchers in Uganda identified a new, considerably more virulent strain of a fungal disease called stem rust, subsequently named UG99 after its country of discovery and year of identification, a pathogen capable of overcoming resistance genes that had, until that point, protected the vast majority of the world's wheat varieties for decades, effectively rendering large portions of the global wheat supply newly vulnerable to a disease researchers had previously considered largely successfully controlled through earlier decades of careful agricultural breeding work. Stem rust itself is a genuinely ancient agricultural threat, referenced in historical agricultural records stretching back thousands of years, with Roman farmers even holding a specific religious festival, called Robigalia, dedicated specifically to appeasing a deity believed responsible for preventing this particular crop disease from striking their harvests. Since UG99's identification, plant pathologists have tracked its steady spread across East Africa, into the Middle East, and towards South Asia, a region containing some of the world's most agriculturally significant wheat-growing areas, prompting an urgent, internationally coordinated research effort specifically focused on identifying and breeding new wheat varieties carrying genuine resistance to this evolving threat before it potentially spreads considerably further into the world's most productive wheat-growing regions. This ongoing, actively managed threat offers a genuinely sobering illustration of how a single evolving pathogen, given the right combination of widespread agricultural monoculture and insufficient genetic diversity, retains the theoretical capacity to threaten food security across a meaningful portion of the entire global population, a threat researchers continue actively working to stay ahead of through dedicated breeding programs conducted in agricultural research stations scattered across the world. The international response to UG99 offers a genuinely instructive example of modern agricultural science operating at its most coordinated and proactive, with researchers establishing what became known as the Durable Rust Resistance in Wheat Project, bringing together plant scientists from dozens of countries specifically to screen thousands of existing wheat varieties for genuine natural resistance to the new strain, while simultaneously working to breed newly resistant varieties suited to the specific growing conditions of the regions considered most immediately vulnerable to the pathogen's continued spread. This kind of large-scale, internationally coordinated agricultural research effort, largely unprecedented in its scale and speed at the time UG99 was first identified, has since become something of a template for how the global agricultural research community now approaches emerging crop disease threats more broadly, representing a genuine, measurable evolution in institutional response compared to the far slower, more fragmented reactions that characterized agricultural crises examined earlier in human history, including several of the other historical events discussed throughout tonight's episode. Long before Brazil and Colombia became the coffee-growing giants most consumers associate with the beverage today, the island nation now known as Sri Lanka, then called Ceylon under British colonial rule, stood as one of the world's leading coffee producers throughout the mid-19th century, its hillsides covered in extensive coffee plantations that formed a central pillar of the colonial economy. Beginning in 1869, a fungal disease called coffee leaf rust began spreading through Ceylon's coffee plantations, and much like the Irish potato blight and the banana industry's Panama disease discussed earlier tonight, the disease found devastating, near-total success specifically because the island's coffee industry relied almost entirely on a single variety planted across vast, continuous plantations offering the spreading fungus little genuine resistance to slow its advance. Within roughly 2 decades, coffee leaf rust had effectively destroyed Ceylon's entire coffee industry, an agricultural catastrophe so complete and so thorough that colonial planters, faced with the total failure of their primary cash crop, made the historically consequential decision to pivot their now devastated plantations toward growing tea instead, a transition so successful and so enduring that Ceylon, and later independent Sri Lanka, became one of the world's most significant tea producing nations, a national agricultural identity that traces its origin directly back to a coffee blight that struck more than a century and a half ago. Coffee leaf rust never fully disappeared following this historical devastation, and has continued periodically threatening coffee-growing regions across the world in the years since, including a particularly severe outbreak across Central America beginning around 2012 that caused billions of dollars in agricultural losses and significantly disrupted livelihoods across the region's coffee farming communities, offering a genuinely striking, century-spanning illustration of how a single agricultural crisis can permanently reshape not just a nation's economy, but its entire enduring agricultural and cultural identity going forward. This same coffee leaf rust story carries a genuinely interesting scientific footnote directly relevant to the emerging field of plant pathology in the 19th century, since the naturalist Harry Marshall Ward, dispatched to Ceylon specifically to study the devastating outbreak during the 1880s, produced some of the earliest detailed scientific work establishing the fungal life cycle responsible for the disease, research that not only helped the broader scientific community better understand this specific pathogen, but contributed meaningfully to the emerging, still relatively young scientific discipline of plant pathology more broadly, meaning Ceylon's agricultural catastrophe inadvertently helped accelerate the development of the very scientific field that would, generations later, prove essential for understanding and responding to nearly every other agricultural crisis examined throughout tonight's episode. Beginning in the 1860s, French vineyards began experiencing a mysterious, devastating decline, with vines withering and dying across increasingly wide swaths of the country's most prestigious wine-growing regions, a crisis that took researchers years to properly diagnose, eventually tracing the cause to a tiny, aphid-like insect called Phylloxera, accidentally introduced from North America, where Native American grapevine species had evolved natural resistance to the pest over countless generations, a resistance entirely absent within the European grapevine species that had been used for winemaking across the continent for centuries. Phylloxera attacks grapevine roots specifically, gradually starving the plant of nutrients and eventually killing it outright, and because European winemaking relied almost universally on grapevine varieties lacking any natural resistance to this newly introduced pest, the insect spread with catastrophic, seemingly unstoppable efficiency across French vineyards throughout the following decades, eventually destroying an estimated 2/3 or more of France's entire wine-producing acreage, a devastation so complete that it threatened the very survival of French winemaking as a viable industry at all. The eventual solution, discovered and refined over years of desperate agricultural experimentation, involved grafting the traditional European grapevine varieties, prized for their specific flavor characteristics, onto the naturally resistant root systems of North American grapevine species, essentially combining resistant American roots with vulnerable but flavorful European vine growth above ground, a solution so effective that the vast majority of the world's wine industry, even today, continues relying on precisely this same grafting technique, meaning nearly every bottle of European-style wine produced anywhere in the world today grows on root systems tracing their genetic origin back to the very same North American grapevine species that first, accidentally, introduced this near-catastrophic pest into European vineyards more than a century and a half ago. The Phylloxera crisis also produced a genuinely interesting, lasting scientific legacy extending well beyond winemaking itself, since the desperate, internationally coordinated effort to understand and combat the pest during the 1870s and 1880s helped establish some of the earliest formal research institutions and scientific societies specifically dedicated to plant disease and pest management, laying important groundwork for the broader field of agricultural entomology that would later prove essential for addressing countless other pest-related crises across the following century. Interestingly, a small number of vineyards, particularly in a few isolated regions with sandy soil that Phylloxera struggles to survive within, along with a handful of locations that somehow escape the pest spread entirely, continue growing genuinely ungrafted, original rootstock European vines even today, producing wines that winemaking enthusiasts and historians regard with particular fascination, offering a rare, tangible connection to how European wine tasted before Phylloxera permanently, irreversibly changed the fundamental biological foundation of the entire global wine industry. The overwhelming majority of the world's natural rubber, essential for tires, medical equipment, and countless other modern industrial applications, is grown today across vast plantations throughout Southeast Asia, a geographic pattern that might seem puzzling given that the rubber tree itself originates natively from the Amazon rainforest in South America, where it evolved alongside a specific fungal pathogen called South American leaf blight, a disease considerably more severe than anything currently threatening Asian rubber plantations. Early 20th century attempts to establish large-scale, plantation-style rubber cultivation directly within the rubber tree's native South American range repeatedly, catastrophically failed, with South American leaf blight consistently, devastatingly wiping out these concentrated plantations, since the disease, having co-evolved alongside its host species across millions of years within the same native ecosystem, proved extraordinarily effective at exploiting the exact same genetic vulnerabilities the concentrated, monoculture-style plantation format inevitably created. This repeated failure ultimately drove the global rubber industry to relocate cultivation entirely to Southeast Asia, a region where the rubber tree could be grown as an introduced, non-native species specifically because South American leaf blight, tied closely to its original South American ecosystem, simply wasn't present to threaten it there, allowing the exact same kind of vast, genetically uniform plantation agriculture that had proven catastrophic back in the rubber tree's native range to instead thrive successfully across an entirely different continent. Agricultural scientists and industry researchers studying this history express genuine, ongoing concern regarding what would happen should South American leaf blight, or some comparably devastating pathogen, ever successfully establish itself within Southeast Asia's now globally dominant rubber-growing regions, since the same underlying vulnerability, vast plantations built almost entirely around genetically similar rubber tree stock, remains essentially unchanged today, simply relocated, once again, onto a different continent where its original natural predator pathogen happens not to currently exist.